Hi guys, this is a short video on classification characteristics of the domains and kingdoms. So you guys have been introduced to our system or level of the taxa. So remember, it's a hierarchical system, meaning that you start with these broad categories called domains. And then as we move down, kingdom, phylum, class, order, there's more and more stringent criteria that organisms are required to fulfill in order to continue to be classified together. And that's meant to represent that continue or more shared evolutionary history and the closer relatedness of those organisms. It starts kind of broad. So, we are going to be looking at the three domains that organisms can be classified into and the six kingdoms. Please realize that the number of levels and their names are going to explode beyond that because as we get down into phylum, class, order, and we make those characteristics more and more stringent for our classification, our categories get much wider and more diverse. Meaning, you are never going to memorize for me phylums, orders, species names, genuses, any of that stuff. Literally just domains and kingdoms. So our objectives for this video, you're going to differentiate between the three domains and differentiate between the six kingdoms. Our skill, we want to be able to walk away from this with being able to identify what domain in kingdom and organism, nice spelling, Miss Curly, would be in based on how it fulfills the characteristics of life. So should be able to tell you, hey, this thing is a multicellular eukaryotic heterotroph that does locomotion. You'd be like, oh, Miss Curly, that's an animal. And I would be like, yeah, high five. All right. Quick thing about our current system of classification that we need to embrace as scientists ourselves. And this is a little bit of an understanding on the nature of science. Everything that we talk about, and I mean absolutely everything with classification, is to the best of our knowledge, meaning that this system of classification is based on every observation that has been made to date and trying to put those organisms into a system of classification. Please realize that there are so many organisms on this planet that have not been fully studied, those that haven't been discovered yet. So, this is an inherently imperfect system based on the fact that we have only the data available to work with. But this is a nature of science piece. One of the things that science is constantly seeking to do, and this is our second bullet, is we always go back and re examine past conclusions. Just because we have this kingdom called animals today and we have these criteria doesn't mean that we're etching that into a stone tablet and we're saying this is the way it is forever and ever at all. We're essentially saying, based on what we see, we think this group makes sense. But we are always looking for more information and always willing to go back and re examine those groupings and those past conclusions. And that's just what it is to be a scientist. So, in terms of our three domains, these ones are nice and easy for you. Domain is all about cell structure, which means you literally, if we were to find an organism out in nature and you didn't know what it was and you wanted to try and classify it, one of the first things you would do is look at its cells underneath a microscope. If you identify that that cell contains a nucleus, then congrats, you have found yourself something that belongs in the domain eukarya. Literally anything that is made of cells with a nucleus goes in the domain eukarya because that domain contains every organism with eukaryotic cells. So we notice the names pairing up ever so nicely. You actually have two domains for your prokaryotes. So prokaryotes, remember, are cells without a nucleus. Pro means no. So again, you take that same tissue sample, look at it underneath a microscope, and you're like, oh, I don't see a nucleus now. Well, congrats, you have found yourself a prokaryote, which again are your bacteria. Now, why do I need two? Well, so what I've got down here is a beautiful picture. This is a phylogenetic tree, so basically a mapping of evolutionary history based on genetic changes of your three domains. You will notice that way back, early on in the development of life on this planet, your bacteria and your archaebacteria and your eukaryotes branch. So that's what that split right here represents. Meaning bacteria separated off early on and became their own distinct grouping of prokaryotes, whereas archaea actually evolved together a little bit longer with modern day eukaryotes like plants, animals. And so they're going to have a little bit more structural similarity in terms of their proteins that they have inside their cells, their cell wall compositions. They're going to be a little bit more molecularly similar to your eukaryotes. It is because of all of those molecular similarities at the most basic level that we actually differentiate between these two groups. If you're thinking to yourself, hey, Miss Curley, do I have to know what all of the molecular differences are between bacteria and archaea that make them different domains? No, you do not. You just need to know that there are two domains with bacteria in them, and they are both prokaryotic. Their names are bacteria and archaea. If we take this domain level, we remember this system works by kind of moving down. So that means that we have a bacteria, archaea, and eukarya as our first levels of classification. And then as we move down to kingdoms, we get more categories with more sort of contingent principles that you need to fulfill, or characteristics is a better word than principles, characteristics you need to fulfill in order to continue to be classified together with another organism. So we notice that bacteria only contains eubacteria, archaea only contains archaebacteria, but eukarya actually has 4 groupings. So we have plants, animals, protists, and fungi. One of the things I did here for you guys is remember that Latin and Greek are heavily used in the scientific vocabulary. So you'll sometimes see that the kingdom plant is also called plantae for the Latinate. Animal is often called animalia for the Latinate, and protist is often called protista for the Latinate. Fungi, no Latinate, just becomes fungi. So these are 2 names for the exact same thing. In this classroom, you are more than welcome to use whichever you would like, plant or plantae, does not matter to me. All right. In terms of characteristics of our kingdoms, so we now want to dig into those 6 kingdoms. We start with our eubacteria. I told you guys that we needed to know the characteristics of life because this would help us with classification since we use these to kind of figure out how things are related to one another. So you'll notice that on the side in my purple column on the left are characteristics of the kingdom. So what characteristics identify a eubacteria? Well, it has to be prokaryotic. Important cell structure. It has a cell wall and it uses a special protein called peptidoglycan to actually build that cell wall. Cell wall composition you're going to see is actually helpful for us for classifying organisms. Peptidoglycan is a carbohydrate. You do not need to memorize the structure of it. You just need to know that it is a carbohydrate found specifically in the cell walls of eubacteria. Eubacteria are always unicellular, but they can be autotrophic or heterotrophic. We can contrast this slightly with archaebacteria. Remember that these groups are pretty closely related in terms of what they look like, how they obtain energy, but it's really those molecular differences, so the protein structures, carbohydrate structures down at the very, very cellular level that are going to differentiate between the two. So we notice similarity, still prokaryotic cells, but now different cell structure. So I'm only asking you guys to remember one example of a molecular difference between the two. And that is that if there's no peptidoglycan in the cell wall, so archaebacteria actually use a different carbohydrate called pseudomurin. You do not need to memorize that one, but they essentially do not use peptidoglycan. That is only your eubacteria. They are still unicellular and they can be autotrophic or heterotrophic. So this closes out our prokaryotics. So now we get into the ones you are probably more familiar with, and those are your eukaryotics. So what makes something a plant? Well, it has to be eukaryotic. It has a cell wall, but that cell wall is composed of a carbohydrate called cellulose. Now, cellulose you are familiar with because if you are writing on a piece of paper, congrats, my friend, you are writing on cellulose. Cellulose is what actually makes up the cell walls, and it's what you see in barks. It's what your paper is made out of, your paper towels in your home, and it's this really rigid carbohydrate found in your plants. Plants also contain these complex internally organized structures called chloroplasts. Now, you were probably familiar with chloroplasts from back in your middle school days when you talked about photosynthesis, because that is the job of a chloroplast. It is a specialized structure inside of a eukaryotic plant cell that does photosynthesis. Now, we had said that eubacteria and archaebacteria can also be autotrophic, which means they also can do photosynthesis. The difference here between prokaryote and eukaryote is going to be the chlorophyll, so that green coloring that allows photosynthesis to occur by capturing energy from the sun, would not be organized into a chloroplast if it's a bacterial cell. Inside a bacterial cell, this chlorophyll would just be floating around in the cytoplasm. So if I were to look at an autotrophic bacteria underneath a microscope, the whole bacteria would just look green. But if I look at a plant cell underneath a microscope, I notice these dark green dots throughout that represent my chloroplasts. All plants are multicellular, and as we just were mentioning with chloroplasts and photosynthesis, they are all autotrophic. They all make their own food from non-living sources. Animals, probably the easiest one because the one you guys are most familiar with. You are an animal, and everything else that we consider an animal is classified as such because they are eukaryotic. So look at their cells under a microscope, you'll see a nucleus, but we have no cell walls or chloroplasts. You are always multicellular and always heterotrophic. So you have to consume your food, and as do all other animals. Protista, so our third of our 4 kingdoms in our eukaryotes. Protista is honestly the kingdom that if we were to reclassify one and reorganize it, this would be the one that we would have to do, because Protista contains all eukaryotic organisms that are unicellular. So basically, if you go out into nature and you find a unicellular organism, so it's made of one cell, but it has a nucleus, well, it can't be a bacteria. So we basically just throw that organism into kingdom Protista, which means we end up with kind of a big diversity in terms of our other categories. And we see that with cell structure. So there tend to be two subtypes of protists. They're what are known as the plant-like ones. So an example of a plant-like one would be a euglena. Euglenas are green and they do photosynthesis and they have a cell wall. But then we also find some animal-like protists, and this would be like your amoeba. Amoebas have no distinct coloring. They have a cell membrane that's flexible on the outside, and they actually do this engulfing thing where they literally fold their bodies around something they want to eat and suck it into themselves. It's very cool. And so we see plant-like and animal-like in our protists, and they can be, as we said, autotrophic or heterotrophic. So again, the tricky thing and the thing to point for here is if it's eukaryotic and unicellular, it's a protist. And last but not least, we have our fungi. Fungi are eukaryotic. They do have cell walls, but the protein that makes up their cell walls, I keep saying protein, the carbohydrate that makes up their cell walls, sorry guys, is called chitin. Now I know you're going to look at that word and be like, it's chitin, Miss Curley. No, it's not. It's chitin. So chitin is the name of the carbohydrate that makes up their cell walls. The vast majority of fungi are multicellular and they are all heterotrophs. So one of the biggest differences or the trip ups that people get is between plants and fungi. They are both multicellular and they are both going to be eukaryotic. But remember that your fungi are heterotrophs. They actually consume dead or decaying organism, where your plants are all autotrophs. So I know that was a little bit of a crash course in characteristics. I did want to throw these up here. So a couple cool things if you wanted to do a deep dive into some of these diversity that we see here. So if you want to look at some of your archaebacteria contain organisms that are known as extremophiles. Phile as a suffix means loving, and extreme. So extremophiles are extreme condition loving bacteria. So if you were to find about bacteria that live on hydrothermal vents at the bottom of the ocean, that is an archaebacteria. They love extreme conditions. So there's a video on halophiles and is specifically looking at the pink lake of Australia. It's very cool. Then you have slime molds, protista. You can learn a little bit about those. And last but not least, this one I think is pretty cool, the cordyceps fungus. So you can see a parasitic fungus in action. So last but not least, check out the cool things. I think they're very interesting, fun little short 2-minute videos on your diversity. But quick thing that we want to make sure we did in this video is differentiate between our 3 kingdoms. So we should know what makes something in domain eukarya, how about bacteria and archaea, and then what are the differences between the 6 kingdoms. What we will start practicing doing in class together is, again, I'll give you characteristics and you would have to tell me what domain and kingdom the organism belongs in. Feel free to come back and watch this video as many times as you would like and email me if you have any questions. Thanks, guys.